Good morning, everyone. It's Thursday, November 5th. Welcome to Brainome's webinar. This is part of our Benefits and Applications series. The topic today is Find the Needle in the Haystack. My name is Lynn Chase, and I'll be your host today. I'm the VP of Product Management here at Brainome. Today we've a really fabulous speaker, Bertrand Hérissoux. He's the CEO here at Brainome. Bertrand's a veteran tech entrepreneur. He started his career by doing machine learning for speech recognition way back in the 1990s. I think that was before it was cool. And after many years in the semiconductor industry, he went back to his roots after he met Brainome's co-founder, Gerald Friedland, 5 years ago. Together, they started Brainome, and they did that with the intent of fundamentally changing the way we do machine learning. Bertrand holds a master's in electrical engineering and computer science from the University of California at Berkeley. Today, we're doing Find the Needle in the Haystack, which is a very special class of very important problems on which Brainome does very well. But we've also got going on in parallel a series of talks about the core technology. And the next one that's happening is on Tuesday. That's CoreTech 301. But CoreTech 101 and CoreTech 201 are archived on our website if you want to watch those as well. CoreTech 101 is a conceptual overview of what's going on with the innovations, the mathematical and algorithmic innovations that are the basis of what we're doing at Brainome. CoreTech 201 does a deep dive on the math. So that's a really good one if you really want to know exactly what's going on mathematically. And then CoreTech 301 does a review of all of that, but then runs through 3 really interesting examples and use cases. Also, you can join us next week to look at how Brainome works on time series data. All of the details on all of these events are at our website, and you can sign up for any or all of these at the URL listed here at the bottom of the screen.
Thank you, Lynn.
Before you get started, Bertrand, I'd just like to invite you all to put questions in the Q&A channel during the conversation, during the presentation, and then we'll be able to do Q&A at the end. Take it away, Bertrand.
Great. Thank you, Lynn, for the introduction. And today we're here to talk about finding the proverbial needle in the haystack in data science. And really, when we talk about that, there's a couple of classes of problem that we really need to define to see what we're talking about here. The first type of data set that we look at, what we call the wide and shallow data sets. So first of all, what we're talking about, the type of problem that we tackle with Brainome's dimension platforms are labeled classification problems. So all these data sets that we're talking about, really, you can think of them as in a tabular format, sort of in a giant Excel format in this particular case, where in the wide and shallow data set, you're talking about tens of thousands of features or columns. We also refer to them as attributes. And the shallow part comes from the fact that you have only typically a few hundred instances or samples of data available for these particular problems. They're not inherently very large data sets. I mean, these data sets typically are in megabytes, but there's a complexity level, the amount of compute power required to find or to create adequate model is actually very, very large and staggering. Typical problem that you encounter that tend to fit this these wide and shallow characteristic would be genome mapping problems, for instance, used to predict cancer, where your target is trying to predict a particular form of cancer or a disease for that matter, and every sample is a human being whose genome has been sampled. Similar example would be molecular compounds used to predict drug efficacy, which is how if you can anticipate just based on the molecular composition of a drug how well that drug is going to perform on humans, for instance. Almost very actual type of problems as well would be industrial food design, where, for instance, if you're thinking of plant-based alternative to meat, and you're trying to formulate a new recipe, if you will, and you're going to have thousands of columns of varying bits of information on how it's done. and how well it tastes. And your target function in this particular case would be whether the consumer actually likes the product or not. So typically, what you end up with these kinds of problem is that you're trying to use machine learning, not necessarily to create a model, although sometimes you do care about the model, but really to gain understanding as to what features are important in your to arrive to a prediction. It really boils down, getting your problem down to just a few attributes. The second class of problems that we look at when we think, when we talk about finding a needle in the haystack is what we call wide and deep data sets. Now, these are very, very large data sets. They're typically the gigabytes for tens or hundreds of gigabytes even of data sets. Again, we're looking at tens of thousands of rows, of columns in there. But now we're having hundreds of thousands or millions of instances, of samples of patients, depending on what you're doing. For instance, if you're looking at, how common is a disease in particular patient population, and that's used for drug design. So these would be, for instance, database coming from the NIH with millions of anonymized patients. And in this case, your columns are really just all the possible conditions and disease that anyone could get, and so as a result, every single person really only has a few checks in there, and these tend to be very sparse, data sets, because most of the columns or most of the data points are empty, and only a few are really filled with people matching criteria of the disease. Similar types of problem would be, for instance, in the ad tech world, looking at consumer behavior to, for the purpose of predicting, what ads to show them or what product to show them, for likelihood of them to buy it. And same thing, you're recording thousands, if not tens of thousands of bits of information on your web user. And you're trying to do a prediction, which is, am I going to click on this ad? Am I going to click on that product to buy it? And so these data sets, which are typically very sparse, are also what we call highly unbalanced, which is when you're looking at the distribution of likely to click or unlikely to click, or whether a person is going to has a disease or not, you realize that 95 or possibly 99 or more percent of the data set will not click or doesn't have the disease, and you only have a small percentage of them really that have the disease. But sorry, that have the disease. Sorry. So what are the existing approach to solving these kinds of problems? And the current approach really is a custom solution, and it's basically relies on human expertise, a combination of human expertise and breaking down your problem into smaller chunks that you can actually analyze. You end up basically using solutions such as the cloud to now spin out hundreds or thousands of instances solving smaller pieces of your problem. And typically, after spending a lot of money and time finding local solutions, if you will, you have to bring back all these bits information and now remerge them into the final, the actual model and information that you're trying to get. It requires, it's typically, it takes usually a long time. It's not terribly predictable. It's very custom, which means if your problem sort of changes, you have to go back and start reprogramming the code behind it to actually solve that problem. It's expensive. The alternative approach is what we call direct calculation, where instead of breaking down your problem into smaller pieces, you pretty much take the entire data set as a whole and try to, solve it. And until now, I mean, most techniques would have been essentially brute force, which, if you look at it from a compute requirement, would be very expensive. Basically, it would have taken a typically months or years of compute power to solve any one of these problems. And so what we do, is really a new idea. We don't have to. We're able to essentially train directly from the data by using the measurements. So how do we do it, exactly? So what we do is we use the power of measurements before you do any form of training, to use something we call generalization measure, to really quickly figure out what combinations of parameters or features, I should say, can correctly predict the outcome of your problem. Now, this talk today is really not meant to be very technical. It's more of a higher level, and I really encourage you to look at our core technology series, 101 for the concept, and then 201 to get more information on the deeper math that we use here. But I can probably explain this in fairly high level just to give you an idea of how these algorithms actually work to solve these very typically compute intensive problem, but we solve them very elegantly and quickly. So if you've seen Cortec 201, this is really just a quick reminder of what we do. What we do is we calculate something that we measure something that we call generalization. And generalization is really, we define generalization very roughly, and this particular formula is really for what we call a balanced binary classifier, which means if your data set is actually perfectly balanced, you have just an equal proportion of we'll get the disease, not get the disease, or we'll click on the ad, not click on the ad, which actually is not the case most of the time, but the formula gets more complicated afterwards. And what we do is the generalization is basically we build this we have this concept of memory equivalent capacity, which is how many decision point for a particular type of model. So it'll be different whether you're using neural network or if you use decision tree. How many decision point will it take to actually memorize the data set that I have? That's I'm not going to get into detail, but that essentially gives you sort of a upper bound of what the model size. And then what we look is using this how many correctly classified instances do you actually get, and that ratio, the more if you're able to correctly predict a larger number of outcomes and your model gets smaller, your generalization gets higher. You basically are figuring rules that are really good and if you compare things, something that usually typically is more general tends to perform better on unseen data. And where this comes, that number is really useful in terms of figuring out whether or not in comparing models, is by looking at the absolute value of that generalization factor or measurement. We're able to tell whether or not you need additional training data or whether your model is purely memorizing. We also call that overfitting. Or if basically your model is generalizing, which means you're actually learning a rule, which is obviously the intent of building a model in machine learning, is to have the model learn rules of your data. Now, in practical terms, in the case of finding a needle in a haystack, what you're trying to do is you're trying to find which feature, or typically set of features, that's usually how it goes, are useful, actually are the important one to find the outcome. Now, in pretty much every problem, regardless of the number of attributes that you have, it's, especially if you have tens of thousands of attributes, it's very, very unlikely, actually pretty much impossible, to have every single attribute contribute to the final answer. And typically, it's really only a small subset that tends to influence your outcome. So what we do from an algorithmic point of view is we're going to iterate through the list of each attribute. And we start by looking at the first attribute, and we calculate what generalization that particular attribute brings to the model. Now, we do this before doing any kind of model training. It's purely a measurement on the data, and we look at alternative types of models. In our case, we're going to look at neural network or decision trees, and we iterate through, the first one, the second one, all the way to the nth one. Could be 1,000, 10,000, 21,000 in case of gene expressions, for instance. And we pick the one that has the best generalization because most likely that's the one that's actually the most important. And then we repeat that for the second and the third most important, etc., by basically calculating the generalization on tuple and triples and quadruples, etc. And we repeat. And at some point, what's going to happen is that as you add additional features to that initial combinations of features that we believe matter, at some point your generalization it's going to stop. Basically, you're adding an extra feature. It's actually going to increase your model complexity, but now you're not you're not generalizing anymore, which means that new feature you just added really doesn't bring anything new to the problem. And then you stop. At that point, you basically are done. And notice you haven't built a model yet. You just have you just have selected the attributes that matter. And why this is important is because if you were to brute force this problem, the number of combination you have to look at would actually be 2 to the power of n, which is an astronomical number, which pretty much would take an infinite amount of time to just brute force. The algorithm we use has an upper bound of what we call order of n squared, which is the worst case would be, if you had 1,000, if you had 1,000 attribute, it would be 1,000,000 comparisons to do. But the reality is, because only a smaller subset of attributes typically matter, we have something that's called n choose 2, which tends to be just a few times the number of actual features that you have. And so this algorithm performs extremely fast, even on dimensionality, which are very large. If you go on our website and you try the demo or register and try to sell the enhanced version of the demo, you can see on smaller data sets what attribute ranking, which is this feature we're using in these finding needles in the haystack problem, really looks like. And what it does is, we're taking the data set, and after measurement, what we're going to do is the platform is going to essentially rank all the attributes that you have in order of importance. And this is an example here this is a credit prediction data set. You're trying to see if every person in this every row is an applicant, and you're trying to figure out whether or not they're you should be issuing them a loan or not. And in this particular case, after measurement, it's going to tell you that the credit history is actually the most important. It's not surprising, because credit history is itself, a number that typically agencies use, but also number of dependents and how long they've lived at their residency, whether they have, but there's a few other things in there. And it's going to recommend you that in this case, it says you could just use credit history and get a really good prediction. That would be probably the most general. So typically, as a user, you would say, well, I can I can just select credit history here and then select everything else and try to build a model. But you could also see how things impact, things are impacted if you add number of dependent, if you had your, how long you've lived there. And very quickly, you can see, you can trade off basically some accuracy for generalization. But what's important is we get this information, which is of attribute ranking, which is exactly what we're doing in these needle in the haystack problem, where you immediately see what features actually matter to the outcome of your for the outcome of your problem. So let's take some very specific examples of things that we've worked on with partners. And this is an example of cancer genomics that we work with a research laboratory down in Los Angeles. And this particular researcher was looking at detecting ovarian cancer. And so they had a data set, one of these needle in a haystack data set, and this is a wide and shallow. It's wide because each column here represents what we call gene protein expressions. 21,000 columns with only 584 patients. And here the class, the target, what we're trying to predict is whether the patient does not have cancer, whether it has a non-malignant cancer, or if it has a malignant cancer. Obviously, something very important to try to detect. And in under 2 minutes, Dimensions create essentially a 2-neuron model. It's a very, very small model, if you think about it, that actually can predict with 100% accuracy whether a person has one of these, falls into one of these three conditions. It's extremely powerful if you think about it. This is, 2-minute time running on a single NVIDIA RTX 280. These are for the techies in in the audience. And what's important here is, this is a very small amount of compute time, and we're able to do this very, very rapidly through our measurements. Now, beyond creating the model, what's really interesting is that the model is one thing, but this actually points, the system will actually show you what genes are important to the prediction. In this case, I think there was a couple dominant gene, and then there was a series of 10 that was also suggested as secondary, which to the scientist actually is quite meaningful. It's not just a black box. Basically, it gives you true understanding of what's going on. Second example that we did, which is the other type of problem, which is a very this is the needle in a haystack problem with a very wide and very deep type of data set. This particular case, it was database coming from the NIH with 2.4 million patients on it, and 20,000 columns, each of them corresponding to potential disease conditions that a patient could have. Very sparse, very, very sparse data. And here the researcher was trying to figure out, so by picking particular disease, figure out what underlying condition would actually result in this particular disease. And again, this it gives you some insight on the biomechanisms which are at play in these particular problems. And so each row here is each row is a patient. Very, very sparse data. This particular database is almost half a terabyte. I mean, this is really a very large data set. And for the first type of problem they were looking for, it took us about 5 minutes to find 23 factors that would predict the particular disease they were looking at with about 99% accuracy. And maybe not all these factors actually matters. I mean, there might be some inaccuracy in the database and other things, but this narrows down your problem very, very accurately, and then lets the researcher do their work afterwards. Very interesting problem. On the same data set, they actually were looking for other things, and for a different disease in about the same amount of time, not as good that time around, but different problem. 62 factors predicting with 92% accuracy some other type of disease. These are very, very complicated problems. And what I want to point out is that we did not design this software to solve specifically that particular kind of problem, but this is what measurement does for you. It's a systematic way of looking at the data that can now solve problems on just about any dimension without the A in this particular case. An interesting thing we've been also working on with some of the partners is to look at food design. And a lot of the prepared food product we buy at the supermarket is really the result of industrial food design. It's not some chef in a kitchen designing it. It's really designed by expert teams that include chemist and bioscientist and of course chef, but there's a lot of technical things going in there. There's hundreds of parameters, if not thousands of parameters coming into the design, lab formulation, ingredients, coloring, flavoring, etc., cooking recipes, what temperature, moisture level, duration, and ultimately physical attributes, what's the texture, the color, the smell. But what really matters is whether or not you and I actually end up liking the product and, we'll buy it. And traditionally in these kinds of problems, which again are very, very wide, but kind of shallow because each formulation really, every time you do a new formulation, it costs money. So you're not going to do 500,000 tests on this one. You may do a few hundreds at most. So traditionally, every group would be responsible and try to optimize separately, the lab formulation and the recipe and things like that. But using our system, there's no more, you don't have to, you can just optimize your problem globally and find actually what really matters in there in really one shot. And there's really no more dicing and slicing required in this one, except maybe to eat that burger that's over there. That looks pretty good. So what's the bottom line with what we're doing? Applying a measurement-driven approach to attribute selection. I mean, basically, is the fastest way. It's lightning fast. It's a lightning fast way to be able to solve these kinds of problems. It's very powerful because you don't need to have experts go break your data set in smaller, manageable problems and then have to put it back together. You can literally process this all at once. We're able to handle very large problems. As I said, we've done data sets that were over 500 gigabyte, with like sparse matrices, for instance. And lastly, it's accurate. So we've had, working with different labs, we've actually had people give us practice problems to say, hey, try on this. And we were able to validate that our results, are exactly, what people would have expected using, the more laboriously intensive techniques that they're used to, that would be obtained at, much greater cost and much longer time. And that's it for today. Lynn, back to you.
Thank you very much for that very interesting presentation. We do have a number of questions coming in from the participants, and I'd like to share those with you.
Okay.
Sandeep is here, and he would like to say this. He says, it seems as if you're saying that there's a way to dramatically change how much time and money it takes to do things like detecting cancer and designing drugs. But the numbers you're presenting in terms of speed and accuracy just seem kind of incredible. What are the limitations on where I'd be able to get this kind of speed and accuracy?
The numbers are incredible. Yes, it is really it's a completely different approach to tackle these problems. Maybe the part I don't make very clear here is that 99% of the work that happens is just measurements, and measurements are very fast to do. You're not doing any training, you're not doing any model building, and for us, the model building and the training is a one-off. You do it once you've done all that pre-processing with measurements, and so you are correct. The time saving, the cost saving is tremendous. There is no fundamental limit to the dimension of the problem that you have. We've never encountered problems that had more than, I think, 24,000 features is the max that we've seen. I suspect at some point there's some impracticality in terms of what people do. In the case of things such as cancer research, obviously we're not going to bring cancer research down to hours overall. The computational step that would normally take weeks, months, or possibly longer, can be indeed shrunk down to days. I mean, that's really what we're talking about here. It's a tremendous improvement, but it doesn't take away obviously the rest of the work of cancer research, which people call the wet lab, where you're actually running experiments and what comes afterwards. So yeah, we're definitely speeding up tremendously the what they call the dry lab, the computational part of the discovery process. Hope that answers your question.
Yes, it appears that that's good. We have another question here. This one's from Rajesh. He says, your definition of generalization, which seems to be the key idea in how you do measurements, that definition included a reference to something called memory equivalent capacity. What is that? I've never heard of that before.
So the memory equivalent, let me go back to the formula I think that I have over here. Okay, so the memory equivalent capacity is actually a concept that was introduced by Gerald Friedland, our CTO and co-founder. And we actually have papers on that. You can actually look on our website. We have information on that. And what it is it's the first of all, it's a number of decision points that's required to essentially purely memorize the data set. But that number of decision point that's required to be memorized is topology dependent. So if you're targeting, say, a neural network, you're going to get a different number that, for instance, if you're targeting a decision tree or an ASR or an forest or something like that. So it's a measure of how much data it requires. I'm sorry, how large your network needs to be to memorize it. That's really what it is. Now that's prior to doing any learning, obviously, but this is a rough computation, and it's a very important number. It's quite fundamental to a lot of the work that we're doing.
Got it. Thanks very much. Mohammed would like to know, what's the biggest data set you could use this attribute selection approach on?
Well, the largest that we've tried is 0.5 terabyte. that's the largest that we've run. But again, we haven't seen really any fundamental limits to what we have. At some point, you're going to need a larger, more memory and more disk space and everything. But the math itself is not bounded and not limited to a particular problem size. We've run data sets with tens of millions of data of rows, and we've done other problems with as many as 24,000 columns. The largest is actually one of these example of disease, trying to figure out disease, which was the order of 0.5 terabyte. That's the most we've tried. But if you have some very large problem you'd like to try in our system, we'd be very happy to help.
Great. There's one more question, and I think we have time for one more question. Sarah would like to know, you gave some examples of your technique working on very large data sets that are very sparse. How does that work? How do you get that to work?
Well, again, when you're looking, the sparsity of the data actually is handled extremely well by this generalization factor that we have here, and the math behind it really does not care whether the data is sparse or not sparse. It's really the beauty of the measurements. It's probably not the right venue today to go into much more detail. I would strongly encourage you to look at our Cortec series, especially 201. I think gets deeper into the math. But yeah, it works extremely well on sparse data.
Great. Well, I want to encourage everyone to, if we could go to the last slide now, Bertrand.
Yes.
I want to encourage everyone who has remaining questions to contact us. You can email us at support@brainome.ai. If you haven't seen the Cortec 201 webinar yet and you have detailed mathematical questions, please do go watch that webinar. And if you still have questions after that, email us at support@brainome.ai and we can get on a Zoom and have a detailed conversation with you. Next Thursday, we're going to be talking about applying the Daimensions software to working with time series data. And this is especially useful in designing health tech devices, wearable health tech devices, and also in the financial markets, working with financial markets. So come on back on Thursday to talk to us about time series data. And next Tuesday is our Cortec 301, where you can also have time with Gerald Friedland, our CTO and the inventor of all of this great stuff. And he's going to go through a bunch of use cases and examples. So again, thank you so much for joining us today. If you haven't done so already, please go on over to the website, sign up for free, and use our software. And if you have any kind of remaining burning questions, email us at support@brainome.ai. Thanks, everyone.
Okay, thank you.